All right, everyone, let's do some Chapter 3 review slides. Yeah. Okay, human digestive tract. Remember, it's good times, right? Really, we're looking at, for each thing, what's going to start breaking it down. So, salivary glands, we're really only concerned about carbohydrates there, right? Your salivary gland is going to start breaking down your carbohydrates, right? And then esophagus, nothing's really happening there, not till you get into your stomach. And then we've got a lot of mechanical digestion. We've got some hydrochloric acid, your duodenum or duodenum, which is connected to your liver and your gallbladder, going into your small intestines when we're going to start absorbing things, and then your large intestines. And then it's going to exit. And remember, it's a tube, right? It's one giant tube. And it doesn't really go in. It goes in and out, but it doesn't go into the rest of your body unless it's absorbed. Okay, let's talk about carbs. The journey of the carb, right? Starting strong in the mouth. We're chewing, mechanical digestion, chemical, salivary amylase starts to break down into polysaccharides, okay? Mini saccharides. Then we're going to break down into disaccharides and monosaccharides. Once we get into the stomach, we're really not breaking down carbs anymore. Your amylase is inactivated by the massively low pH of your hydrochloric acid, right? Then you're turning the corner into your duodenum, which is connected to your pancreas, and that releases pancreatic amylase, salivary amylase, pancreatic amylase. Again, breaking down your polysaccharides into monosaccharides. The small intestines is going to use that chemical digestion, and it's going to break it down. There's some more enzymes in there, maltase, sucrase, lactase, and it's going to break down into monosaccharides. Large intestine will primarily be for indigestible fibers. Here's a picture of that. Good chance there's going to be a picture on the exam. Some matching, okay? So you're going to have maybe some matching with, right? This is the mouth, and then match the example of what's in there. Salivary amylase, again, starts to break it down. Nothing happening here. Stomach, not really doing anything for the carbohydrates. Not till you get into this duodenum and your small intestines, where you're going to break it down and also absorb it. The microvilli, remember the little fingers that are increasing the surface area? We have two things going on. Well, three, really. We've got SIGLT, right? Our sodium glucose transporter, right? So we got sodium. Where do we get the sodium from? Your favorite transporter and mine, the sodium potassium pump. Okay, so we get it in through SIGLT. We get it out through GLUT, the glucose transporter. This is facilitated diffusion. Facilitated means you need a facilitator, you need a friend, you need a buddy to move your carbohydrates, your monosaccharide sugars in and out of the cell. Pretty pictures first, then my picture, right? Here's our friend SIGLT, sodium glucose transporter. Also, remember, we're not eating the glucose. It would just be like if I worked at Krispy Kreme, I'd eat all the donuts. No, it does not use glucose for energy, because otherwise we'll just eat it all. Just a side note. Okay, and then so we're getting it in, sodium glucose transporter. Here's our sodium potassium pump. We're leaving through a GLUT. Okay, same thing the other side. Sodium glucose transporter, sodium potassium pump, GLUT transporter. Okay, same thing. Here's my picture. Looks exactly the same. Exactly the same. Here's SIGLT coming in strong, right? Sodium, glucose. Glucose is leaving through GLUT, and it's just going right into your capillary. It's just moving right in, right? Because it's oozy, leaky, and your monosaccharide is real small. So it just goes right in, down its concentration gradient, not even it's just diffusion, right? Right in here. Your sodium is also doing that. Your blood is moving towards your liver, okay? This was brought to you by today's sodium potassium pump, brought to you by sodium potassium pump, okay? And then your GLUT transporter right here. Remember, can't use glucose, uses glutamine as an energy source, okay? You should have this written on a colored piece of paper. Okay. Dehydration synthesis. How do we break down these polysaccharides into monosaccharides? Dehydration synthesis. It's a condensation reaction. It removes water, okay, and joins two monosaccharides into a polysaccharide. This is how your monosaccharides are formed. We are breaking them down by hydrolysis. We're adding a water molecule, and we're breaking that chemical bond and converting it into a monomer in the process of digestion. Okay, hydrolysis adds water and breaks the chemical bond. Dehydration, condensation reaction, removes water and creates the bond. Okay, looks like this. Monomer, 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 water reaction. Look at that. This is hydrolysis. We're hydrating it. This is, right, we're hydrating it with water. Look at that. Dehydration, hydration. We're adding water. We're breaking it apart. We're dehydrating it. We're losing the water. Okay? The G words. Okay, remember our little box for the G words? Glucose is the primary fuel. This is your breakdown of a monomer into glucose. Glycogen, the storage of that glucose in your liver or your muscles. Glycogen is the storage flour of starch. Starch flour of storage. Glycogen. Okay? Glucose, glycogen. Glucagon is the hormone that raises blood sugar. Insulin is the hormone that lowers blood sugar. Okay? It just doesn't start with a G, so it's not on here. But glucagon is the opposite of insulin. Okay? Glycogenesis. Genesis meaning born. Making glycogen, starch flour of storage, from glucose. Making genesis, glycogen, the starch flour of storage, from glucose, the sugar molecule. Okay, the fun continues. Glycogenolysis. Lysis meaning to break. Breaking down glycogen into glucose. So at some point, you're going to need that back. So this is when we break down, lyse means to break, glycogen back into glucose so you can use it. Okay? Coming in strong at gluconeogenesis, making new glucose from your fats or your proteins. Okay? And then glycolysis. This is the breakdown of glucose into energy. This is glycolysis Krebs cycle electron transport chain. Okay? Here's the picture. Remember? Glucose is the box. Glucogenesis is the making of glycogen, this starch box of storage. Okay? Glycogenolysis is when the worker breaks it back down into glucose. You put it into the bloodstream and you're going to get glycolysis. Gluconeogenesis is making your glucose from something else, from your proteins or lipids. Okay? Homeostasis. This is blood sugar level rises. You eat a donut. Your beta cells in your pancreas release insulin. We have a picture of that. Right? You release insulin. The body cells take up glucose and perform glycolysis. Glycolysis moves on from here. When you have enough, your liver takes up the glucose and stores it as glycogen. Starch flour of storage. Okay, the blood glucose level declines because it either gets used or stores as glycogen. And then you're like hungry again. You have lower blood sugar. Okay, you have low blood sugar. It's 3 o'clock in the afternoon. Your blood glucose level falls. Your alpha cells in your pancreas are like, hey, can you release some of that glucagon? Because we could use that glucogen. We could use some glucose again. So glucagon is released. Remember, it's the opposite of insulin. And your liver breaks down the glycogen and releases glucose, raising your blood sugar, keeping the whole thing in homeostasis. Okay, here it is with a cookie in the middle. It's not a cookie. That's your pancreas. Those are the cells in your pancreas. Okay? Glucagon or insulin, depending on which way you're going. You release insulin because you have a lot of glucose. It goes ahead and makes glycogen, right? You release glycogen and it goes ahead and breaks it down to glucose. When it does this, it raises the blood sugar. When it makes glycogen, it lowers the blood sugar. Okay? Same thing with the muscles, right? You release insulin, it goes into your muscles. You make glycolysis. Okay? You release glycogen here, and it makes glucose and raises your blood sugar. Okay? Just go through that a few times and make sure that you understand it. Here's your pretty pancreas and your duodenum. Two of my favorite organs. Okay, so you have glycogen. Also, let's remember that your pancreas, why it's one of my favorite organs, because it's both an exocrine and an endocrine gland. What? Yes. It's an exocrine gland because it puts these digestive enzymes right here into the duodenum. It's an endocrine gland because your alpha and beta cells, release insulin into your entire bloodstream. Look at this. It releases it just into your blood and says, there you go, insulin. Right? So it's an endocrine and an exocrine gland. We'll come back to that. Okay, we are going to review how that insulin got into your bloodstream. It's our old buddy GLUT. Again, glucose transporter. Yes, it is. Yes, it is, sir. Okay, so you have a high level of glucose, and GLUT is like, you better bring some of that in here. Why is it going to go in here? Because diffusion, right? It's just going to come in through your glucose transporter because there's a low level of glucose in here. Now, what's crazy, remember that when we talked about the small intestines, we said it doesn't use glucose. It doesn't use it. It uses glutamine. Well, fun fact, your pancreas is like, yeah, give me all the glucose. I'm going to use that, and I'm going to make me some ATP, right? I'm going to make a ton of ATP. What? Okay. So your glucose molecule comes in here via GLUT, mitochondria, powerhouse of the cell. We're going to dive into that in a minute. And it makes a bunch of ATP. Okay, awesome. What does that do? That increase in ATP opens a potassium. This is potassium. Opens a potassium gated channel. See, this is a gate. It looks like a gate. Come on, that's a gate. Right? When you increase the ATP, it opens this potassium channel. Who cares? Between that ATP and this potassium, you get a voltage difference. Okay? It creates an ion concentration voltage difference. That opens a voltage gated calcium channel. Yes, it does. A voltage gated calcium channel. I know you are at this point like, please tell me why I care. I'm getting there. I'm getting to the exciting part. Your calcium channel adheres here to your insulin and creates exocytosis, and it barfs out insulin into your bloodstream. What? Review. Glucose transporter, mitochondria, ATP, bunch of ATP, opens your potassium gate. That creates an ion. Concentration, voltage gated. Because of the voltage change, that opens a calcium. Voltage gated channel, that calcium binds to your insulin in little packages here, and it exocytoses it, aka spits it out into your bloodstream. And that is endocrine because we're putting it directly in the blood. Okay? We're not putting it next door. We're putting it all over the blood. Okay? This is in your islet of Langerhans. You got to say, well, you have a German accent. Okay? Yes, maybe, possibly. This looks exactly like mine. Come on, totally the same. Glucose transporter in here, in here. We're making a little glycolysis, which we're going to talk about in a minute. That mitochondria makes a bunch of ATP. That ATP opens a potassium channel that depolarizes, making the voltage gated calcium channel open. Your calcium comes in, binds to your packets of insulin, and your insulin exocytoses, out your insulin into your bloodstream. Those pictures look exactly the same. Boom, boom. Totally the same picture. Okay, so you got insulin in your bloodstream. Who cares? Glycolysis cares. How are we going to make ATP from that? That, my friends, shall be the next video.